that reverse transcription is a repair mechanism for chromosomal DNA. But the mainstream of molecular genetics is still committed to the central dogma. There is no such thing as reverse transcription from RNA to DNA. In 1970, when they detected biochemically that there is a reverse flow of genetic material, they didn't give up the dogma or even try to change it. Instead, they called it an exception to the central dogma of molecular genetics and explained it by postulating the existence of retroviruses. Zengers. Excuse me, but I thought that the field of retrovirology had started as far back as 1911, with Peyton Rous and his experiments with chickens. Rous surgically removed cancerous tumors from chickens in his lab, ground up the tumors, fed them to healthy chickens and observed that the healthy chickens who ate ground-up tumors grew tumors themselves. He concluded that the tumors may have been caused by an infectious agent being transmitted from the sick chickens to the healthy ones. Dr. Lanka. No, it was only in retrospect that he was cited as the one who was dealing with retroviruses. What Peyton Rous actually did was he inbred his animals so heavily that the genetic material from the different strains he used to breed became more and more similar. When the animal's genetic materials become too similar to each other, then even more genetic material is interchanged between the chromosomes than happens normally. Often in inbred animals or plants, on 2 places of the chromosome's genetic material in between got lost. Then you will see the characteristic chromosomal damages in inbred animals, plants or human beings, resulting in disabilities which are well studied. So, because Rous's chickens were so heavily inbred, they had a high rate of spontaneous cancer induction. The results from this research were not cited for more than 20 years. Later, some people tried to speculate about them. In the late 1960s and early 1970s they started to think about this because molecular biology took over modern medicine, and argued against the existing body of knowledge, of facts, that cancer is caused by infectious entities, by viruses, or mutations, or viruses causing mutations. They ignored the fact that cancer has something to do with oxygen deficiencies, which had already been established by Otto Warburg's research. Warburg had received his first Nobel Prize demonstrating how a cell is able to produce much more energy than in the process of fermentation, using oxidative respiration. And he had received his second Nobel for proving that cancer is characterized by the process of fermentation, that oxidative respiration is not taking place in cancer. And this has been just ignored. So in 1970, when they proved that reverse transcription does happen and they discovered the enzyme reverse transcriptase which does it, they wouldn't give up the dogma. They changed it slightly, and said there is an exception, and that it was associated with the existence of a new class of viruses called retroviruses, which they cannot prove exist in other ways. When I was absolutely sure about everything I've told you so far, I went public. I was invited to a lot of conferences on marine biology and biology, and at every conference I presented my own data. I used every opportunity to speak out against HIV, and I quickly learned that because I was taking away HIV as an explanation for AIDS and was not able to replace it with something else, and not being able to explain what's going on under the label HIV, it forced me to watch out and find those people who were able to explain what's going on. In the beginning, of course, some of the publications of Peter Duesberg helped me a lot because he was an authority who questioned a lot of things, and that helped me. I translated some of his articles into German and published them in a small publishing house. But then, with time, I learned about other specialists, among them Heinrich Kremer, the well-known German medical doctor, former medical director of the Federal German Drug Abuser Clinics, who helped me to understand what was really going on, because he was in charge of the introduction of hepatitis B vaccine into Germany, and used it in his patients. Dr. Kremer checked out the hepatitis B vaccines on the market. He found that the American vaccine, hepatitis B vaccine, was produced with the sera donated by men in the gay scene in New York City between 1978 and 1980. So, as he knew, there was a lot of sex going on in a minority of these men, and therefore they had had a lot of sexually transmitted diseases. So he was afraid of using this vaccine, and instead he used the French vaccine, which was produced from blood donations by the general population in France. But in 1983 the German government forced him not to use this vaccine anymore. They said the French vaccine is poisoned by the AIDS virus, at the time when nobody was positively speaking about an AIDS virus, but the American vaccine was okay. He knew, or he was warned, that this had nothing to do with the science, but it had to do with the fact that the German medical system, in parts of Germany, is virtually a colony of the American system. Soon after, in 1984, he was told to deliver frozen blood samples of his patients to Berlin, to the newly founded AIDS Center, to be tested for the AIDS virus. Before he let his blood out, he checked what's the evidence for the accuracy and reliability of the HIV antibody test, and he realized that this test is not able to detect the virus. It is not able to say yes or no, you are or are not infected. It is only able to say that you have a higher or lower amount of antibodies. That's how the HIV antibody test was and is designed. Zengers. It's my understanding that when you have an antibody test that is actually useful, like the antibody test for syphilis, you get a high or a low antibody reaction, and it's a certain multiple of how many times you dilute the original sample and still have the reaction. Therefore you know not only that the infection is present, but also how well the immune system is responding to it. Dr. Lanka. I'm absolutely sure that no antibody test in medicine has any absolute meaning. Especially in HIV antibody testing, it is clear that the antibodies that are detected in the test are present in everybody. Some people have them in higher concentrations, and some in lower concentrations, but only when you reach a very high level of antibodies, much higher than in any other antibody testing, are you considered to be positive. This is a contradiction in terms because in other antibody tests, the lower your level of antibodies, the higher your risk for a symptomatic infection. But with HIV they say you are positive only when you have reached a very high level of antibodies. Below this level, you are said to be negative. Zengers. So, this is what Dr. Roberto Giraldo was talking about when he spoke to HEAL in San Diego. He said that when they do the HIV antibody test they dilute the sample to 1/400th of its original strength, and if they didn't do that, all the samples would test positive. Dr. Lanka. That's it. How ridiculous. Dr. Creamer knew this already by 1984. He was very worried about the fate of his patients, because in 1984 the politicians asked him to put these already stigmatized HIV-positive patients into quarantine, which means to separate them from the other ones. He said no, because there's no infectious entity out there. He knew everybody who went through chronic active hepatitis, or had the hepatitis B vaccine would test HIV positive. So he knew that there is no infection in his hospital. He informed the mass media, who went to his hospital to inform themselves, in great detail. He told them all the evidence. And the very same journalists, in talk shows in Der Spiegel, one of Germany's largest and most popular magazines for example, published just the contrary. So he knew that it was intentional from the very beginning. They played war. They all wanted to have a blood and sex plague, contrary to the evidence which he presented to them. So he knew that AIDS was built upon misconceptions. He was dealing at the top political level. They told him, off the record, that they knew they didn't care. It was about how to deal with the drug problem, and with the homosexuals. They even tried to kill him, and this didn't succeed. He had a good intuition, and got out of his car before they